All right, guys, we're back. Function of membrane proteins. As you can see, there are a lot. Some are hormone binding sites. For example, the insulin receptor sticks on the outside, and when insulin comes to the cell, it triggers downward communication. Immobilized enzymes with the active site on the outside. Example, small intestine, they have embedded enzymes on the outside to break down food. Some proteins are cell adhesion to join cells together, so make them stick to each other. Some are involved in cell-to-cell communication. For example, receptors for neurotransmitters at synapses. Some are simply channels for passive transport, which we'll talk about in the next section, to allow hydrophilic particles to cross the tails by facilitated diffusion. So it's like a straw through the membrane, a channel, and then something that's hydrophilic can fit through the straw without being repelled by the tails. And then they have pumps for active transport, which use ATP energy to move particles across the membrane. Again, we'll talk all about that next section. Okay, so types of membrane proteins. We have the integral proteins, which are hydrophobic on part of the surface and therefore embedded in the hydrocarbon chains. Some are transmembrane with hydrophilic parts projecting through the phosphate heads. We'll see pictures. Peripheral proteins are hydrophilic on their surface, so they are not embedded. They stick on the heads. Most are attracted to the surface of integral proteins. Others have a single hydrocarbon chain to anchor them to the membrane surface. Finally, cholesterol in membranes. Now, this is found only in animal cell membranes. So if you ever see a vegetable product that's labeled as cholesterol-free, well, no duh, because plants don't have cholesterol. Okay, cholesterol is a type of lipid that is a steroid. It's mostly hydrophobic, so it's attracted to the tails. But one end has a hydroxyl group, that's an oxygen and a hydrogen, that is hydrophilic. So it's mostly in the tails, but it's attached to a phosphate head in the middle. Cholesterol controls the fluidity of a membrane. So it prevents tails from packing together and becoming too solid, but it also restricts molecular motion and becoming too fluid. So it just gives it the right amount of fluidity. But let's look at a fluid mosaic model, the current model. So starting with left, we have phospholipids with the blue phosphate head and the two fatty acid tails. Below that, that's a peripheral protein. In this example, it sticks, it's got a little bit of hydrophobicness that allows it to insert into the tail, but it's mostly on the outside. Then we've got some integral proteins. One is a channel protein for passive transport. That's like the straw that I said, where it would allow hydrophilic proteins to pass through. The other is a carrier protein, which we'll see more of next section, which pumps, it opens and closes using energy to allow things through. We have cholesterol there on the bottom right of the model in yellow. You can see that it's mostly hydrophobic, so it's in the tails, but it's got that little tiny hydrophilic part that allows it to be inserted at the heads. And then we have a peripheral protein with a glycoprotein. We did not talk about glycoproteins, so we don't need to worry about that. And if you want to go to this slide share, it goes into these notes a little bit more in detail. All right, guys, next we'll talk about membrane transport. That's it for today.